Go.
Good afternoon, my name is Jelena Simeunovic and today I have very important news for you. First one is the bad one, and that is that we have waited for far too long and done far too less to address the climate crisis that we are facing. But since after the rain always comes the rainbow, or in my case it comes the sun, since I will be talking about PV power forecast, there is also good news. And the good news is that if we act immediately and on a large scale, we can still address this issue. And we are going to do that by increasing the number of renewable energy sources into the power grid. But in order to do that, we must have accurate PV power forecasts such that we can balance this grid. And having accurate PV power forecast is actually a difficult task. Let me show you why. As you can imagine, when clouds are covering PV station, PV power production is going to be low. And since they are constantly moving, shame of them, they are making our job much more difficult. As they are constantly moving from one PV station towards another, they are going to affect them in similar manner, which means that PV power production at this station, which might be in Bern, and this one, which might be in Lausanne, are going to have similar patterns. So what if we revert the problem? What if we actually use PV power production data to track clouds? Because by observing different systems in time, we can find similar patterns. However, in the real life, we don't have two PV stations, we have many more. We have, well, in Switzerland, 100,000 PV stations. So how are we going to address this issue? And here is where the graphs come to rescue. If we observe each PV power station as node in the graph, then PV data is lying on top of these nodes. And then the links connecting these nodes are actually going to be used to pass pattern information, cloud information from one node to another. So this is how we are going to have accurate PV power forecast. Phew, I already feel better about news today. How about you?
Hi everyone, and today I will introduce about the story about vaccination and our bodies. This is MS. It's very tasty, right? Actually, it's very similar structures with virus. This is the peanuts inside and the outer layer chocolate layers. And its peanuts actually can represent the virus genome, which contains a lot of the nucleic acid information, and is the crucial part that leading you have some symptoms of infections. And this is the outlayers, and which can represent the virus capsid. And you can imagine that virus also need a clothes to cover them. And what we can do to manipulate these outlayers is as the Moderna did in previous. And I think people in this room all take the third shot for Moderna vaccination, right? And what Moderna did, actually, they first carefully observe the outlayers of the virus and take the note, oh yes, all code orange, size 2. And then they will carry these informations. When you inoculate Moderna on your arms, and Moderna will circulate in your bodies, and they want to find the communities. And then Moderna will encounter with an excellent immune cells, which we call it the dendritic cells. Dendritic cells is excellent decoders for the informations. So Moderna will tell dendritic cells, hey sir, information, notice, code orange, size 2. And when the dendritic cells got the information, he will say, oh yes, got it. And dendritic cells will bring this information back to the command centers. And in the command centers, actually, there are abundance of the immune cells ready to fight against a virus. And in this case, dendritic cells will code B cells like soldier B, remember that code orange, size 2. And so B cells will reply that, oh yes, sir, I will remember the code orange, size 2, attack. So at that moment, B cells were equipped with the powers to fight against the virus. When they circulate in the bodies, B cell will encounter with the virus again, and they will produce a kind of protein which we call antibodies. Antibody will infiltrate the virus, smash them, and digest them. And this is how vaccine training our immune system protect us. Thank you.
You know when you are in a lab? Yeah, okay. You know when you are in a lab and you're just fascinated how cell molecules reacting to the surrounding signals communicate and cooperate among each other to create all existing life forms. Inside every living cell, whether it's plant, animal, or bacteria, there is literally an ongoing concert under the conducting baton of environmental stimuli dictating rhythm to gene orchestra to play all kinds of symphonies so the cell can dance. As it is an orchestra, it means that every gene has its turn to play and some will initiate other genes' turn. For example, regulatory proteins encoded by some genes will trigger other genes to play their keys and notes. So I will take this cell culture into the incubator, but before I shake a bit an Erlenmeyer, and it's like I'm hearing this symphony of life. When every cell divides, going through life phase and it dies. But before it dies, cell is dancing through life stages. 'Cause regulator proteins are playing their genes like piano, yes, yeah. Yes, they can change, they can play, they can change. Cellular shape, cell response, they can change if they properly fold, if they know how to hold. Promoter sequence is a place to enhance or suppress gene expression. Yeah. Cardiac cell, muscle cell, neural cell. Signaling path will ring genes, jingle bells, and there are a million different melodies to which cell can dance. What is yours? What is yours?
Have you ever imagined the importance of colors in our life, and to be very honest in the science, to be very precise in the cancer biology or tumor microenvironment diagnosis? You might have heard about the colorful festivals like Holi, for that matter, like good over evil, and celebrating different colors and happiness in India. Second example would be the Carnival Festival in Italy, or Painted Red, that, as we all know, that is Tomatina in Spain. Hence, let me take you to the world of colors in the field of cancer immunology. So in cancer immunology, we have two different terms, which is, one is antigen, that is the foreign particle, and the second term is antibody, that is the idea what detects the antigen or the foreign particle. This figure showcases the antibody with the fluorochrome. So these green color small balls are the fluorochromes, which are the colorful objects which help in detection of the tumor or cancers. And this picture showcases the antigen-antibody interaction. Let me quickly show you two images, one with the keratinocytes of our skin, and the other the nucleuses and the nuclear material of nucleuses which are stained with DAPI, and this is stained with the molecular biomarker that is keratin 10. We can diagnose cancer, or we can diagnose tumors with the colors, and that's super fascinating for me. I'd like to demonstrate one thing here. If this is the T cell, and the biomarker for T cell is CD8 positive and CD4 positive. The fluorochrome label antibody will signalize the T cell in any patient sample which has a cancer or which has a tumor. And for the nucleus, the DAPI will be staining in green or in blue color. Hence, we can diagnose with the confocal microscopy different colorful images, and which can lead to diagnosis of cancer in the early stage or maybe in the aggressive progressive stage. In the second figure, we can stain different cells with different antibodies labeled with different fluorochromes, and hence this is the colorful world in the cancer immunology. Thank you.
See those guys? They represent the proteins that are working inside our cells. And as you can see, they are very diverse and they have many different kinds of functions. And what is fascinating is that even if they have been done probably forever, we still discover the function of some of them today. And for example, this one, my favorite one, his name is DDI2, and it's what we call a protease, which explains this word. This word, it's the protease domain of DDI2, and it should allow him to cut other proteins in order to make them active. And so far, DDI2 has one known substrate that is known as NRF1. And when NRF1 is active, it will promote the transcription of some of those guys that have very specific function, and they will combine the strengths in order to build the biggest protein complex of the cell that is called the proteasome. And this complex has a very important role inside the cell. It will promote the degradation of non-functional protein in order to build new one. Now, imagine that this guy is non-functional. Then you will have accumulation of useless protein that will inexorably lead to the death of the cell. Sounds terrible, right? Except if we are talking about cancer cells. Some cancer cells, they produce a lot of protein, which lead many of them without proper function. And that's why they need to be degraded by the proteasome. Then it's also why, for some cancer, medicines target directly this complex in order to make him inactive and kill cancer cells. But do you remember the role of this guy? Yes. It promotes activation of NRF1 that will increase the transcription of proteasome subunit. Then, of course, it's annoying if you want to make this guy inactive. And that's why I only work on this guy in order to find a way to inhibit him and avoid apparition of adaptation to current treatment. So, of course, the sword is very important. But do you see the pursuit of DDI2? It required a structural domain. And I discovered that when you remove this structural domain, then DDI2 cannot cleave NRF1 anymore, even if the sword is still present. And I'm sure there is other mechanism that explain cleavage of NRF1 by DDI2. And that's why we need to pursue our investigation on this swordsman in order to make him a new therapeutic target for cancer therapy and see what surprises is still reserved to us.